Why are conservation efforts failing genetic diversity? Imagine a world where species seem to be thriving because their numbers are growing, but underneath, their genetic makeup is weakening. This is a common problem in conservation today. Conservation efforts often focus on increasing the number of animals or plants without paying enough attention to their genetic health. Genetic diversity is the variety of genes within a species, and it is essential for populations to adapt to changes and fight off diseases. When conservation strategies only aim to boost population size, they risk creating populations that look healthy but are actually vulnerable because they lack enough genetic variation. One reason conservation efforts struggle with genetic diversity is that they tend to measure success by how many individuals there are or how wide an area a species covers. But a big population can still have very low genetic diversity if it started from just a few founders or went through a bottleneck. A bottleneck is when a population shrinks drastically, causing many genes to be lost. These populations may seem stable but are actually at risk because they cannot adapt to new threats. Another issue is that conservation plans often focus on ecological factors like habitat protection but ignore the genetic side. Many conservationists do not include genetic knowledge in their plans. This means they might overlook the importance of gene flow, which is the movement of genes between populations, or the need to protect unique genetic traits. Without this focus, some populations become isolated and lose their genetic variation over time. Funding and political interests also play a role. It is easier to get support for saving a charismatic animal or protecting a beautiful habitat than for genetic research, which is less visible and harder to explain. This leads to less investment in genetic monitoring and management. Focusing only on specific genes, like those related to immunity, can be problematic too. Sometimes, managing just a few genes ignores the overall genetic health of the species. Genes work together in complex ways, and protecting only certain ones might cause the loss of important adaptive traits. Environmental factors and population structure also influence genetic diversity. For example, inbreeding depression happens when close relatives breed, which can reduce fitness, especially in stressful environments. On the other hand, populations connected by dispersal can maintain diversity despite local inbreeding. These dynamics make it hard to spot genetic problems without long-term studies. When genetic diversity is lost, species become less able to adapt to threats like climate change or new diseases. This can lead to species extinction and reduce the resilience of entire ecosystems. For agriculture and food security, losing genetic variation means fewer options to breed crops or livestock resistant to future challenges. To improve conservation success, it is important to include genetic monitoring in plans. Protecting multiple populations across a species range helps preserve local adaptations and gene flow. Using genetic tools can identify populations with unique traits that need special attention. Sometimes, introducing individuals from other populations, known as genetic rescue, can boost diversity but must be done carefully to avoid risks. Combining genetic data with ecological and demographic information allows for better, more balanced conservation strategies. In the end, conservation efforts often fall short of maintaining genetic diversity because they overlook the complex genetic processes that keep populations healthy and adaptable. Shifting focus from just counting animals to caring for their genetic health is key. Improving genetic literacy among conservationists and balancing short-term gains with long-term evolutionary potential can help protect species for generations to come.